Afternoon session here. So I have the pleasure first of introducing Professor C.Y. Wu from the University of Miami, where he serves as the chair of the Department of Chemical, Environmental, and Materials Engineering. Dr. Wu is the inaugural department chair. His teaching and research interests range from air pollution control, aerosol science, environmental nanotechnology, dust control, to engineering education. A fellow of American Association for Aerosol Research, he has published 170 plus referee journal articles with an H index of 53 and over 10,000 plus citations. His research has resulted in 9 US patents and 4 pending applications. We're grateful to have you today, and welcome. Thank you so much.
Thank you very much. Very glad to have the opportunity to share our lessons learned in the past few years regarding environmental aerosol sampling of infectious SARS-CoV-2. And I want to address issues by answering 3 questions. What is the goal of the study, and where do we do the study, and how do we do the study? It's not moving. Okay, yes. So the first important question is, what is the goal of the study? Because that decides the experimental design. And since the start of the pandemic, there have been a lot of debates about the role of aerosol transmission. And as pointed out earlier, in the beginning of the pandemic, in the early 2020, and we were advised by CDC and WHO that, oh, aerosol is not the route, and you don't need to wear a mask. And as long as you are 6 feet away from the source, then you should be maintaining 6 feet of social distancing. That should be fine. But to a lot of aerosol scientists, that doesn't make sense. But in order to answer that question, you needed to prove the role of aerosol in the transmission. And for that, you do need to have 3 things. And you needed to have the emission source, the infected person, and you need to have another recipient to receive the agent, so you get sick, right? And then you also need to have an aerosol in between to show the connections of these 3 components. In order to conduct this kind of study, you do need to have either a human volunteers to be infected. That doesn't happen in the United States, or you have to use animal studies. And still, that's very limited. So what about the next level? The next level of the study is, well, we can exclude the recipients, so we don't infect volunteers. And for this case, we needed to have the emission source who is sick, and then we also need to collect the aerosol. So for this, we conducted the studies in hospital, and we put our samplers within certain distance from the infected patients. And then for this kind of study, you typically have the known source. You know, you have a sick person over here. And our objective was to match the genome of the nasal saliva sample with the air samples. And in addition, we need to cultivate the virus in cells to prove its viability. This has to be done in the BSL-3 facilities. And also IRB and the biosafety requirements for sample transport and storage are very complicated. And don't mention about the virology expertise to the analysis costs a lot of money. So this can be done easier than the previous one, but still it's very challenging. And that's why most of the studies you can see in the literature focus on just the aerosol part, excluding the source and the recipients. And there are all different kinds of samplers used to collect the air samples. And once the samples are collected, you process and they're analyzed typically by RT-PCR. Okay, so this is very good for observing the trend, similar to the wastewater surveillance. You can do that. The information over there is useful for that purpose. However, in order to talk to the medical experts in WHO and CDC, they would not use this type of data because detection of RNA cannot be used for assessing risk associated with the transmission of virus. That's because abundant RNA fragments are produced and continually shed, but they are not infectious. And therefore, they are not really of concern from the infection perspective by the medical experts. And the other issue is very often we don't know if there is an emission source is present in the environment, and that causes an issue. So hopefully with these 3 slides, you can see, the objective of the study would dictate what kind of sampling strategy you need to have in order to answer the question, and they are different. The next question I want to answer is, where do we do the sampling? Well, so that is to answer, where does the transmission occurs and where is the hotspot? So in the beginning of the pandemic, healthcare facilities were the epicenter, and therefore there were a lot of studies conducted in the healthcare facilities. And after the PPE protocol was implemented, then the infection case in the healthcare facilities among the healthcare workers reduced, and therefore they were no longer a hotspot. So what do we do the next? Okay, so after the reopening, there were a lot of concerns about the public spaces. Can I go to the gym to do the exercise? Can I take the public transportation? And how about sending the kids to school? Is that safe, right? So there were a lot of studies conducted in these public spaces. And there are a few issues associated with this kind of study. The first one is we really don't know if there is an infected person, so we don't have the known source, right? Different from the previous case. And during the pandemic time, mask mandate further reduced the possibility. So what? Well, in that case, the low probability requires a huge number of samples over a very long period of time, very expensive. Let me show you on the right-hand side of the results over here. This is from University of Michigan. They collected 256 samples, and only 4 out of the 256 were positive, so about 1.6 positivity rate. This more or less follow the case numbers in that area, so can be used, but this is very low. So it's really a probability issue. And the other thing is when you have a low concentration in this kind of a condition, and also the coexistence of other viruses and atmospheric oxidants will make it almost impossible to determine the viability of such collected samples. And this also requires, so for this kind of condition, it really requires a different monitoring strategy compared to in the known source cases. Okay, now how about the next? Well, so the next one is the sampling in the public space doesn't really provide a lot of positive samples, but still there were a lot of infections going on. So where is the transmission occurring? Where is the hotspot? The same question, right? So we turn the attention into residential space. And why is that? That is, we realize in the residential space, there's no mask, no social distancing, and very often no constant ventilation. So I'm showing you the results of recent studies actually conducted in 2021. And the concentration is the y-axis, and the x-axis are primary and secondary locations. The primary location is the self-isolation room where infected individuals spend most of the time. And the secondary is outside of the isolation room, but in the same building, okay? So you can clearly see from the results over here, there is no difference between primary and secondary location. So what does that mean? Well, sorry, go back. Right. So this says that the viable SARS-CoV-2 aerosol can be transported to other space in the same building. So isolation room with a good intention in residences is good for isolating human beings, but not necessarily virus. So this has a big implication how we should set up the mitigation strategy to better protect the occupants inside. Hey, and we conducted another large-scale meta-analysis of SARS-CoV-2 concentrations versus location. And here we have for healthcare facilities and also for residences. You can, and also for primary and secondary. You can see from this large data set that the primary location and the secondary location in the residences still no significant difference. And furthermore, the residential primary concentration is much higher than that in the healthcare facilities. So risk is higher in the residence compared to the healthcare facility. And one big difference between these two type of location is the ventilation rate, air change per hour. In the hospital, it's required to have a 6 air change per hour. In residences, recommended 0.35, and very often you don't really have a good ventilation. So you can see the big difference over there. And I also want to point out there are still a lot of factors to consider for where to do the sampling. And for example, we are advised that you should place the samplers at the breathing zone in order to collect the samples more accurately. But we also know that human thermal plume tends to lift the virus. So the plume actually gets the virus from the immune source higher than the emission point. So that brings a question, is the breathing zone really the best place to put your sampler, right? And then another one over here is sampling in school, for example. In school, it's so large. Where do you put the samplers, okay? And well, we thought about all students would gather at the cafeteria. They would talk loudly, and they don't have mask on, so that would be a good place to do the sampling. On the other hand, the ventilation is really good at the cafeteria in comparison to a classroom. Classroom is much more crowded, and their activities over there would actually be a good place to do the sampling. But if you want to do that, you need to have, say, 50 classrooms, you need to have a 50 sampler. Can you afford that? If not, you're going to choose one or two, and which one or two? That's a low probability issues, right? So there are a lot of things that need to be considered. And I want to point out the need to develop standard protocols for sampling respiratory virus aerosol, which doesn't exist yet. Okay. Then the next question is about how do we do the sampling? And again, keeping in mind that we wanted to efficiently collect the virus aerosol samples and maintain their viability. And during pandemic time, there were so many types of samplers used and no standard. And the issue is that the virus may get inactivated due to desiccation during the sampling on filters. And the other thing is, when you try to extract the virus from the filter, that can also damage the viruses, and therefore you lose the viability. So we wanted to bring in a new device that is inspired by nature. For in this way, we would encapsulate the virus by water vapor or by water and amplify the size for easier collection. And in the typical atmospheric environment, we have the condensation nuclei. They are so small we cannot really see them. However, on a cold day, for example, like today in Arlington, and when you exhale, you can see the white plume. That's because of the water vapor condensing onto the nuclei to make them larger, can better scatter light, and therefore our eyes can see them. This is the same process in the plume from stack or the cloud formation. So we package that into a machine, and we first have the conditioner that is cooling the aerosol particles. And then in the initiator stage, we'll provide the high moisture content to condense the water vapor to make them larger, and therefore they can be efficiently collected for further analysis at the collection plate. Okay, so I want to quickly show you the first study using the H1N1 virus aerosol. The x-axis is the number of infectious virus generated during the sampling, and the y-axis is the infectious virus collected. And you can see that the virus samples, which are the new device samples represented by the blue dots, they are at least one order of magnitude better than the industry standard, the biosampler. And during the pandemic, we also conducted a study side by side of the virus compared with the NIOSH sampler, which is a filter-based sampler. And you can see the virus positivity rate is much, much higher than the NIOSH sampler. So this tells us that the condensation-based sampler is better, much better than the cyclone filter-based sampler. And also it points out the need to develop a standard method and sampler. So the choice of a sampler is critical, and the standard sampler is essential. Now, I do want to use the remaining time to point out two directions I think are important. The first one is about the transmission. As a function of distance, can it be transmitted far away? It's a function of particle size. And therefore, in sampling, if you can sample the particles by size, that will give you the information you needed. So we added a biocascade that are able to separate particles by several size ranges. And we tested it in the laboratory using the human corona OC43 virus. On the right-hand side, you can see the genome equivalent of the OC43 compared with the aerosol size distribution measurement, and they match very well. And on the lower right-hand side, you can see the cells directly inoculated with exposed to the OC43 after 7 days later, and then lose their viability and they float. You can see the difference between these. So this shows the possibility of using such a device to directly expose the cells directly expose the cells to the virus, so you can get the size fractionated information. And the other important piece of information is about point of care detection. Because right now the samples are collected and sent to an off-site lab for analysis. It may take a few days before you can get the results back, right? So at that time, it's already too late to inform occupants whether there is a high risk of exposure. So if you can do the analysis right over there in real time, that would be the best. So this slide shows sliding device that you can have different chemicals at different stages. And when you slide one by one, you can have the sample introduction, and you can have the lysis of the sample, and you can bind the RNA and wash it, and then using the RT lamp to amplify it. So the right-hand side shows the results of such a device for SARS-CoV-2 and for influenza, successfully detecting these two viruses, and in 15 minutes. Okay, so that's all I have, and I want to have a quick summary. I hope I have demonstrated it's very important to identify what is the goal of the study, because your experiment design depends on your goal. And the second is, where are you going to do the sampling? Where does the transmission occurs? Where is the hotspot? There are a lot of questions about this, how to do that. And the next one is the sampling, sampler, which type of sampler, how do you do that sampling, right? And also want to point out, it's very important to develop the size fractionated capability for sampling viable virus. And we need to develop the capability to have a point of care detection right over there. And standard sampling protocol is needed. Choice of sampler is critical. And finally, I had a lot to work with a team of diverse background, with virologists, with infectious doctor, and also with a biomedical engineer in order to make this work, and the physicist. So this is very essential. And finally, just want to acknowledge the financial support, especially the RAPID from NSF. That's how we were able to get the first samples of the viable SARS-CoV-2 virus from air. So really appreciate NSF support from here. That's all I have for today. Thank you.
Fantastic. We appreciate your remarks. And next, I have the pleasure of introducing Dr. Karen C. Van Miller, Associate Professor of the Civil, Environmental, and Geodetic Engineering and Environmental Health Sciences at The Ohio State University. Dr. Van Miller directs the Indoor Environmental Quality Research Group, which addresses emerging concerns with the built environment. Work aims to improve understanding of chemical and microbial processes indoors that impact health, especially childhood asthma. Thank you so much. Now we look forward to your remarks.
Thank you so much for having me. Can you all hear me okay? Great. Really excited to be here today. Thank you so much for the organizers for putting on this great event. Let's see. Oh, there it goes. Oh. So my name is Karen Dannemiller. I'm an associate professor at Ohio State University, and I do direct the Indoor Environmental Quality Research Group. And I'm really excited to talk with you all today about some of the work that we've been doing, looking at dust as another matrix to potentially monitor for respiratory illness such as COVID-19. So when the pandemic hit, we really wanted to think about the building scale. So as I mentioned, we're in the indoor environmental quality research group. We wanted to say, is there a monitoring strategy that we could develop to specifically target the building scale? We've heard a lot about the awesome work going on with wastewater monitoring and the huge successes that have been there. And that's really great for these larger scale areas. It can be done at the building scale as well, but oftentimes you might have difficulty accessing somewhere where you can collect a representative sample of that specific building. And on the other end, and I think of this as kind of a spectrum, right? You have individual testing, which everybody loves to go get tested every week, right? No. But you can kind of get higher resolution data of specifically who's infected. And so we wanted to look at, is there a monitoring strategy that we could develop to target this intermediate scale? And you can think about where this might be useful. In our case, on a college campus in dormitories, but also in places where you might be especially interested in knowing what's going on, such as nursing homes, prisons, and a lot of other locations. So this is everyone's favorite picture of a sneeze, right? And we wanted to think about, in the indoor spaces, where is this virus going? And we've heard a lot about the little particles that are resuspended in the air for a really long time, and those are likely responsible for transmission, right? But there's a wide range of particle sizes in here, and a lot of them are ultimately going to fall to the floor. And so if we think about what these particles look like, in this case of SARS-CoV-2 is about 0.12 microns in diameter, but it's usually not floating around by itself. It's going to be in the droplets that we exhale for breathing, coughing, sneezing, talking, and these can range from about 0.1 to 100 microns in diameter, roughly. And so if you look at where these go, on this graph, you have deposition velocity on the y-axis and particle diameter on the x-axis, both in a log scale. And you can see that there's kind of this dip where those particles that are right around about 1 micron in diameter stay in the air for a really long time. The larger particles tend to end up on the floor. So we said, can we possibly use this information to figure out where the virus is going in the indoor space, not necessarily to study transmission directly, but to figure out how we can do environmental monitoring in these indoor spaces. So again, the goal of this project was really to develop monitoring at the building scale. So the first thing we did was we went into the isolation rooms on campus. So this was in fall of 2020, and we collected air samples. This was a passive air sampler, so nothing as cool as what we just saw, but a passive air sampler here, and surface swabs, as well as bulk dust samples from the isolation rooms on campus. And what we found in this case, and remember this was fall 2020, our air samples were only about 20 or 30% positive. And these were the rooms where everybody was infected. They were waiting out their isolation period. The surface swabs were about 55% positive. On our first pass, the bulk dust samples were 89% positive. And we rapidly could increase that to 97%. Since we've been doing this, we probably are somewhere closer to 100. And so we also had the highest concentration in the dust samples as well. So we have the concentration in red and the percent positive in the blue dust samples. I worked with a lot of folks on the pandemic committees at Ohio State, and one person said, we were talking about pooled samples, and they're like, I wish that we could just have everybody who walked into a building spit into a cup, and then we could later go sequence that and figure out what the cases look like. And I said, well, what do you think dust is? So there's your happy thought for the day that you can take home with you. So after this study, we rapidly scaled this up, and we started running pilot scale work in spring of 21. And this is an example from one of the residence halls on campus. And we saw that the values in the dust over time corresponded fairly well to the known cases in that residential space as well. So we were actually able to see the positive cases and how they compare to the values for the dust. And in fact, some of these were even sequenceable for the variants in the dust. And I'm going to talk a little bit more about that in a minute as well. Some of the cases here, so we were actually collecting the vacuum bags from the custodial staff who were just collecting the bags as they normally did. We would just take them and, instead of throwing them away, they'd give them to us. One person's trash is another person's treasure, and we would bring them back and sequence them. So in this case, this was some of the private rooms where the vacuums were just available for students to use as they see fit. And then the common areas were cleaned by custodial staff. So we were able to scale this up. So we've been running full scale since August of 21, looking across about 50 buildings a week on Ohio State University at the Columbus campus. And we've actually been able to see trends in the dust consistent with what we know for the case data. So this is from fall of 21. And what you can see is that you can see sort of two darker red areas, one kind of in the middle and one towards the end. The interesting thing is this correlated well with the known cases that we had on campus. The red was delayed actually because there was pre-arrival testing. And so actually a lot of our cases were actually removed before they even made it to the residence halls. And we didn't see them show up until a little bit later. When you take a lot of our data, and this is from one semester, and you compare the known case values with the dust data, you do get a correlation. So it's blocked off, but the P is like 0.0004. The R is relatively small, so the data is noisy. And that's because of some of the issues that a lot of people have spoken about earlier about just variation in shedding rates and time in buildings and things like that. But we do get this nice correlation with values in the dust compared with known cases on campus. And we can actually use this to statistically estimate disease prevalence. Because of the current variability in the dust, you're not really going to be able to tell the difference between one case or two cases, but you are going to be able to tell the difference between 4 cases and 10 cases, or at least estimate those types of differences. So based on the viral shedding rate. So we're still working on some of the statistical analysis here. One interesting question is actually which is more important, the number of cases that led to this environmental deposition or just the environmental deposition itself? Does it matter if you had one person spread a lot of virus into the room or 10 people spread the same amount of virus into the room? I think that that's an open question. So this is the work of John Van Dusen, a PhD student in my group. And so we also looked at the variants that we could sequence out of the dust. So the dotted lines are from the saliva testing data on campus. And then the bars are the variants that were detected from our dust data. And you can see they correspond quite well. Unfortunately, the transition to Omicron happened right over our holiday break. And so we're not really able to tease out what that lag actually looks like if there is a lag there and what that looks like. We'll have to continue analyzing the other data sets to look for that. But you can see that there is some association there. We do see maybe what's a little bit of relic RNA hanging around for the Delta that persists on, but we can detect the new variant as well. So what does this mean about the viability of SARS-CoV-2 in the dust? Absolutely nothing. So if you think about what the virus looks like, it has this fragile exterior envelope, this hard protein capsid, and then the RNA in the middle. And we were actually shocked to find the RNA in the dust at all, because if you work with RNA in the laboratory, you know it's extremely fragile. It's one of the hardest things to work with because RNases are everywhere. And the only thing that we can hypothesize at this point is that it must be protected in that hard protein capsid. So for the virus to remain viable, though, that envelope also has to stay on the outside. So we wanted to ask the question, though, can we get maybe some more answers about what this might look like? Can you do next slide, please? So what we did is we used 2 different bacteriophages. Our BSL-3 labs were also very busy. So we used MS2 and Phi6. MS2 is a non-envelope virus, so it just has that protein capsid and the nucleic acid in the middle. And Phi6 is an envelope virus like SARS-CoV-2. And what we found, so this is the work of Nick Nastasi, and we deposited it onto 2 different types of carpet as well as directly onto the dust. And this is something I think people have been talking about a lot in these different sessions. And the interesting thing that we found is that the MS2, that non-envelope virus, persisted longer, maintained more viability in that dust compared to the enveloped Phi6 that has that fragile outer envelope. And this is consistent with other surface types. So if you look at studies that have deposited these things onto like stainless steel or other surfaces like that, that these the envelope viruses can be much more fragile in sort of maintaining the infectivity. So this is obviously not direct data on SARS-CoV-2. This is a bacteriophage, but it looks like the viability will likely decay for these types of viruses based on the data that we have. But I would say this is an area where we could use a little bit more research. Let's do this on SARS-CoV-2 and see what it looks like. It also begs the question about envelope viruses. Should we look at how these envelope viruses on materials such as dust? The interesting thing though is even though the viability decays, the RNA persists. And this data in this particular paper, we just went out for a little bit of time, a couple days. But if you look at the RNA over time, you see it in published data we have out to a month, and we have other data we've done even longer, that the RNA is actually very persistent in the dust. So, and this is if you collect a vacuum bag from building, bring it back to the lab, you don't even need to store it cold, throw it in the corner, come back later, that RNA is still going to be there. It's not necessarily true in buildings, which is probably due to other removal processes. So when we look at our campus data and you see the RNA over time, you do see that decay about 2 to 10 times per week. So that's probably removal from other things like cleaning and people tracking things in, stuff like that. But the RNA is sitting there ready to be analyzed as a marker of what was in that space. We also looked at removal by cleaning methods for both the viability on the left and the RNA persistence on the right, and different methods that we used. So we had our negative control, we had untreated material, as well as vacuum, hot water extraction, steam, and disinfectant. In this case, what people usually call steam cleaning is actually hot water extraction. For this study, we did the hot water extraction as well as like actual steam. We were just kind of curious what that would look like. And you can see the viability does decrease for some of these, but a lot of times the RNA does continue to persist for these different cleaning methods for the virus in the flooring material. So overall, DUST can really complement wastewater monitoring. And this was used on Ohio State's campus during the pandemic, where we were able to really match up our data with that wastewater data. Oftentimes, the buildings would be like 6 residence halls that empty into one catchment site. And they would say, hey, look, here's a signal. What do you see? And from the dust, we could pinpoint 1 or 2 buildings that it looked like that was where it's maybe coming from. All that data is still in the process of being analyzed, but these things are really, really complementary to each other. And I think should be part of a bigger strategy to addressing viral outbreaks in the future. Wastewater monitoring can, of course, address a much larger catchment area in a single sample, and you have some flexibility there and has really good detection values. But the sample collection can be difficult. So if there's a specific building that you need to target, that's where DUST might be able to step in as well. In the end as well, we're talking about a respiratory virus here. So not everyone actually does shed it in feces. And so these things can actually complement each other in terms of what you ultimately want to look for. So overall, this is really a new long-term surveillance solution that can fit into this broader framework of how we might be able to address viral outbreaks in the future. We also have data on influenza I didn't show here, but you can detect that as well. And so there's a lot of things that we can really do to continue to build this out. I had to throw in too, I'd love to continue the conversation with you at Indoor Air. So workshop and symposium proposals are due next Monday and abstracts due December 1st. So this is a fantastic group. I love talking with you all. I'd love to see you all in Honolulu. So thank you so much to everybody who contributed to this project, my fantastic group of students. They do all the hard work in the lab. And of course, NSF that also contributed to this, as well as our other funding agencies and all our great collaborators. So thank you very much.
Okay, so at this time, we're going to open it up for any questions, both for Dr. Dan Miller, but then also for Dr. Wu as well. Yes, please.
So Karen, really nice presentation. So one question I have for you is that could you elaborate a bit more on your method for the RNA assay? So I'm assuming you're using RT-qPCR. Is that the case? And I'm just wondering if you really target a very small amplicon, like a small amplicon size in your RT-PCR assay, you may underestimate the RNA integrity because, let's say, if you do not see a lot of reduction in your RNA signal because of your amplicon size is really small, then you may underestimate the RNA reduction in your carpet compared to the entire virus viability. So that's another question because I'm not so sure what's the decay mechanism for RNA there. So is that going to happen in a specific site, or is that like just once you see a decay, the entire RNA will be decayed?
Yeah, that's an excellent question. So in terms of the RNA, we were using just a typical RNA extraction. We started with a QIAGEN kit and then eventually moved to a Promega kit later in the processing for the RNA extraction. And then we were just using the CDC assay to target SARS-CoV-2. So I think when you measure RNA, you can't say anything about viability because it really matters a lot of time. And if you think about the two graphs we had about the RNA persistence versus like detectable RNA versus the actual viability, they really don't tell you about one versus the other. So really our questions were, is RNA there? Could we detect a virus that was shed into the environment that indicates that somebody was in that space shedding virus, and now we can pick it up and indicate that there was somebody that was a case in that space. Versus like we're not really in this case with the monitoring, we're not necessarily interested in viability as well. So does that answer your question?
I think in part, yes. So back to because one of the conclusion, if I understand your presentation really well, is that you're saying that the RNA is more persist in the environment you are testing versus the entire virus viability. Is that what you're trying to conclude there?
Yeah, the RNA is going to persist much longer than the viability from what we can see with our bacteriophage work. It hasn't been done on the actual virus. And the persistence that we're talking about is in a collected sample. So it appears to be very different if I like vacuum this room and I put the vacuum bag somewhere, and then 2 weeks later I vacuum this room again. The RNA is going to remain persistent and at the same level in the sample I stored, but you're going to get a different value from what you collected here. And that's probably because of different removal mechanisms to be determined. That is that vacuuming, is that people tracking things around, is it sunlight? I don't know, probably not, but there could be a lot of different ways that is actually degrading in the environment versus in this stored sample. So in terms of the RNA persistence, the point there really does persist in like collected samples, and then there is some decay in the environment, probably due to other mechanisms.
Amy Pruden from Virginia Tech. So to follow on that point, so the assay you use to do the RT-qPCR will matter in terms of the primers, how long the amplicon is. Mm-hmm. We've seen this with antibiotic resistance genes where we're curious, how far do they persist through wastewater treatment? Do they survive UV light? And if we have a 1500 base pair amplicon, it'll show more rapid degradation because the PCR reaction cuts off. Like somewhere along that length, there's more probability it's going to be damaged somewhere. So the same probably applies to RNA. So like when we had like a 75 base pair amplicon, it would show, oh wow, that antibiotic resistance gene really persists. So yeah, there's a lot of nuances, but I think you showed that the RNA does persist. That's the bottom line, right?
Mahendra from Clarkson. I have a question. When you're collecting the dust in the vacuum bag, so there are chances that we are losing some RNA over there. So like, are we actually representing the right amount of RNA which we are collecting from the room, or like, is there a specific number that we are losing there and we know those numbers?
That is a great question. So in this case, when we started this project, we talked to environmental health and safety, and they would not let us change any of the protocols that the custodial staff were using, because it was very early in the pandemic and there were concerns, and they basically said you cannot change the risk in terms of what's going on. So we, especially in the beginning, had to just accept the vacuum bags as they were. Other people have asked questions about, like, what about contamination within the vacuum? Well, we're getting typically 1 to 2 kilograms per week from each of these sites of dust. And so hopefully the amount that's like in that same vacuum from in that same building isn't contributing too much to our samples, but absolutely could be. In terms of what's being lost in the vacuuming, so these are, I would say, in some ways I actually like this large representative sample. Oftentimes when we go to a sampling site, we don't have the luxury of like sampling all the surfaces, right? We typically don't actually go clean an entire indoor space when we're doing our sampling. And in some ways, this is like a much broader sample of that entire area of the building. Some of these are whole buildings. Some of these are like floors 4, 5, and 6, you know, just depending on building size. So we for sure are losing some, but I think it is representative of like sort of copies per milligram dust in that interior space.
Hi, Aaron Bivens, LSU. I have a question for both of you, actually. On the one end, we have the, RT-qPCR molecular data. On the other end, we have we need a BSL-3 space. I mean, do you think there's room along that gradient for new approaches where we can maybe get closer to infectious without all the requirements of BSL-3 lab space? Like, are there computational techniques, or maybe we look at a few different analytes that allow us to move closer towards infectious without necessarily all the infrastructure?
I think that's a very good question, but a hard question to answer, because when you wanted to have a viability, yes, you can check some of the analytes that the viable cells continue to release. You can do that, but in order to have the, how do you call that, limited agents, or like SARS-CoV-2, you are required by law to do any processing in BSL-3 facilities. So I don't know at the current moment, how do you overcome that regulation requirement? Technologically, I think it's possible, but regulation-wise, that may be a different issue.
And yeah, I think that there are some ways that we can get around this. I don't think anything will ever truly replace what happens in those BSL-3 facilities. But so for instance, in our study, we used the bacteriophages, which were sort of like the closest we could get without actually using the BSL-3 space. And I think the BSL-3 space is so time limited in terms of what you can actually do in there, that I think it's important to actually use anything we can, whether it's computational or other laboratory techniques, to kind of step up to that, to try to, once we get into that space, be performing the best experiments there that we can. So I sort of see it as along a spectrum, where the BSL-3 space is usually what's going to kind of prove anything related to viability, but there's a lot we can do on the spectrum pointing towards that.
Maybe I can add one more point. I think Karen pointed out an important thing in terms of the surrogate. Like we use the OC43, it's also a coronavirus. It's not exactly the same, but it's much closer. So that's something you can work with at the BSL-2.
Any more questions?
Not to hog the time, but to hog the time. Karen, a question for you. In terms of temporal resolution of this dust sampling, right? Like daily sampling versus, I mean, are you seeing signal increase and decrease on those kind of what's the time scale for those increases and decreases? And is it, I was kind of surprised by it in the wastewater, how fast it moves. And so I'm curious about dust in that same regard.
Sure, we've really just been looking at weekly sampling. So it's hard for me to comment on anything that's shorter than like a 1 week time period. But we do see the weekly sampling contribute. Could you show my extra slide by any chance? I have one, so had to make it count. there are absolutely temporal issues as well. So if you think about what's going on statistically here, on the left, you can see like, here's theoretical cases that are going to peak at different times. And then in the red is sort of your sort of sampling period. It's also complicated by the fact that in some of these times, different cases were going to be actually removed to isolation rooms. So you have some amount at the beginning that's shed into the building. The person is removed to isolation, then comes back and maybe continues shedding. And all of this contributes among these different cases to one composite sample. And then you have to like run the statistical analysis on that to actually figure out what's going to happen. So there is absolutely a lot of variability there in terms of everything that goes into that and the temporal scale. I think there's so much variability among individual people and how much they shed, and then also over the course of their own illness. But in this case, the dust is probably going to tell you kind of like what was in that space at, you know, one given period in time, what was actually shed into that interior space.
And just to note, there is no hogging the mic either. So please feel free to ask as many questions as you would like.
I have a question. So, I mean, how do you put these together? I mean, like, I can see wastewater surveillance as like give you sampling for a broader, you know, like community, right? I can see dust, air sampling, more like distributed, localized. I mean, how do you link those different scales and proposal, I don't know, policy implementation? I mean, just think forward. How do you think about it? I don't know the answer.
I think that is an excellent question. And I think that's one of the next big questions that we actually really need to go after is what does true integration of these systems look like? Because all of them have their own pros and cons and their own strengths of what they can look for. And I think really using them all together is what's ultimately going to get us to the best spot. So in terms of the dust wastewater question, we're still working on that analysis, but we at least anecdotally saw times where we'd see this high wastewater signal from our catchment site with say like 6 buildings. And we could say, hey, look, buildings A and B also had, or building A also had this high signal coming out at that time. So the question is, how do we use it? You know, is it something where maybe we use wastewater for the catchment site and then we collect the dust samples and just run them, you know, when we see them? I don't know. And then how does this all ultimately go to influence policy? How does this work with air samples in individual spaces as well to understand what's going on? So I think it's going to all be part of developing this longer term monitoring strategy that also needs to be able to scale up and down depending on the signal that you're seeing. So there's probably some background monitoring level that would be good to have ongoing. And then maybe you ramp up testing if you start to see signal that you're concerned about. And then how does that influence policy? When do you ask for, you know, different interventions? So I think that's a really important question.
And I think probably in terms of air sampling, I will start from that. It will move forward to say local sensor, because right now, you know, EPA has the PM10, PM2.5 standard samplers for monitoring the long-term air quality. And but at the same time in the market, you can see the local sensor replacing, or now I shouldn't say replacing, complementing what the standard monitors are doing. So I would see, I would envision in the future, there will also be something like that. You can detect the pathogen almost in real time and in the individual space. But at the same time, how do we get there? I think that requires technological advancement in the detection side, but also incorporating all the data that Carrie mentioned. You know, you needed to incorporate the wastewater surveillance, the dust, and also the air sampling, because they are at a different scale. They provide you different information that you can use to create a model to predict the risk, and you can focus on those places where you need to do mitigations instead of everywhere is important, then everywhere becomes not important. Does that answer the question?
We've been thinking, from NSF perspective, we look at these things along a continuum, like we have emergence, transmission, and mitigation, right? So I hear air and wastewater surveillance more on the transmission, but can they go, like more downstream at the emergence, right? I mean, we hear zoonotic transfer, but the way we think about it is like one virus jumping to another animal. Maybe zoonotic transfer is more than that. Can you guys comment on air, water sampling for emergence?
So I think that once again it's putting all these pieces together, and I think the animal question is actually also a piece of this puzzle. We haven't really talked about that too much, but I do think it's also a critical piece. And one of the biggest challenges is to also look for the emerging pathogens that we maybe don't know about, those unknown unknowns, and figure out if there are ways that we could potentially look for those as well. For the things that we know about, we at least can use a lot of these different matrices to be kind of screening and looking for where they might be popping up. We can also use them to look for new variants, which a lot of the nice thing about a lot of these samples is they represent a lot of people all at once. So you're more likely to pick up on new things that are maybe concerning as well. But I think it all goes into this like concerted effort with all these different pieces that are contributing different components to give us the best picture that we can get with the resources we have.
And my take is, like Karen mentioned, this is a One Health issue. We do need to incorporate the animal side. I think there are a lot of experts in the agricultural, domain that we should work with to get this figured out. That would be my answer to this question. But maybe as others in the area that would share their thoughts.
I think I can. Hi, this is Andrea from NYU. I think emergence is one of the most exciting opportunities for wastewater surveillance. I don't know how well it could work with air, but thinking about targeted and non-targeted. So a targeted approach, if there's an organism that we know is an issue that hasn't been in a place yet. And one example is poliovirus detection in New York City, in the counties north of New York City. We had a seminal case, but then we were able to see how it spread. So it was a really exciting application of wastewater surveillance that happened recently. And I think about certain tropical vector-borne diseases that maybe we should start looking for in wastewater in new places that we didn't traditionally have those vector-borne diseases. But then this question of emergence of brand new pathogens is a challenge. I'm curious to know what everyone thinks, but it's like non-targeted analyses that we would need, which is not something we're thinking of routinely in wastewater surveillance right now. But yeah, I'm not sure about the air side in terms of looking at emergence, but interesting as well. It's just challenging, I guess, in terms of the scale, and that you can't sample as large a community. It's maybe the challenge.
So maybe I can share one thing that we did in 2016 before the pandemic. We went to the Student Healthcare Center and do air sampling, and you will be surprised how many types of viruses we will be able to sample over there. But that's a snapshot, and that's not necessarily monitoring a surveillance network, but it does give you the window to observe what may be going on, and then develop a strategy to do the long-term sampling. Maybe healthcare center is one, but you already have sick people over there. How do you do that in the general population? That's a different story, require different strategy. And there we also discussed about, say, doing in the airports, because the airports are where you probably are the first entry point, from other places. But there are, I think, scientifically that's intriguing and should be feasible. But on the other hand, when we approach the airport, they told us, get out of here. If you step in, we're going to throw everything out of it. So that may require some more assistance in order to get this done. I think you need to establish network and connection with people. I think if I remember correctly, Lindsay Ma was able to do sampling in the aircraft because she has been working with that community for a long time. So that gives us the access to the aircraft health and safety issue. I think similarly, if we wanted to work on other area, we needed to really network with them to get to know them. And so we can really work on those important questions.
I'll just weigh in a bit here.
I have a question for Professor Wu.
Go ahead.
Sorry.
I was going to weigh in on the
Okay.
last question.
So Professor, you mentioned in one of your slide where you did like meta-analysis of patients, like the primary, I would say in primary hospital, you have mean value lower as compared to the residency where you have a higher concentration. And you mentioned that air change rate is one of the biggest factor there. But do you also think that like the location of the sampler do also matter over there? And also like, because you have, I think 125 participant in hospital and only 12 in the residency. So we are actually not comparing apples with apple here. So how are we getting this?
Okay, let me explain that. The meta-analysis, we collect data from the literature. We didn't do the sampling ourselves, so need to clarify that. So all the 125 studies were all conducted by others, and definitely you are correct that, hey, is the same sampling location, same type of sampler? Definitely no, because there is no standard protocol, there is no standard sampler. But even with those uncertainties over there, we can still see the drastic difference between a residential space and the healthcare facilities. And we don't have all the information, but based on our hypothesis that ACH is important parameter, so we can see that correlation. Can I say that's the causal relationship? I cannot say that, but there is a strong correlation for that. Hope that answers your question.
Just, I guess circling back to the emergence question, I mean, it seems like a great opportunity to, I know there's a lot of work in this space in disease ecology, right? And looking at where are the conditions right for emergence and then applying our tools in those spaces. I don't know that we have the people in the room to answer, that question, but, and then it seems like there's probably, I mean, it seems to me there'd be some motifs we could search for, right? Like we know SARS-CoV-2 is not entirely novel in terms of its genome. So there may be opportunities where we can shortlist, hey, these are the ones that we expect to be potential players in this space. I think we knew about coronaviruses and especially zoonotic coronaviruses as a potential threat. And then we can search for those specific signals in an environment where maybe we're expecting that emergence to occur. Might be a way to go about having a look at that.
I mean, can you find rare biomarkers, right? Emergence typically are rare events. Like in everything you do, can you get to a point where you like, this biomarker is so rare that maybe it's going to do something? Can you actually decompose the signals to find rare biomarker of rare events?
So I think that's a really interesting question. And so I spent many years studying dust, which, as a kid, I had never thought I would say that, but I've spent many years studying dust. And if you look at the microbes in your dust samples, like 98 to 99% plus or minus, you don't care about, right? They're just there. There are these, potentially hundreds of different species that you're just like, well, they're just there. We don't really know anything about most of them. A lot of them have never been cultured or studied in any in-depth capacity. Where definitely somebody talked about database issues being biased towards clinical samples, and that's absolutely true. That's what we tend to know about. But I think it makes it really challenging in terms of the emergence question, because we have the bulk of what we're looking at, these samples we don't really know much about, and we potentially don't really care much about. And so it's really trying to be able to figure out where's that one that we don't know about yet, and why is it bad? And I think that's a really, really challenging question.
Yes, and forgive my ignorance, but I'm curious about how difficult it is to do that sort of separation in terms of the detection. Is that I'm just thinking about other industries like maybe trying to detect certain pharmaceuticals or special chemicals, right? There are very specific tests that are required to be able to differentiate between things that might be similar or even things that are very different. And I'm wondering when it comes to these biomarkers, like how difficult is it to say, oh, this is something novel and new compared to this is something that's known and common?
I think, and others in the room can also join in who have similar experience in this space, but it really depends what technique you're using. So if you're looking at microbes, for instance, you might be looking at the amplicons, and in some cases, it's just going to be a sequence of DNA or a sequence of RNA, and that might be all the information that you have about it, and you're just like, it's this relatively short sequence. That's all we know. If you use other techniques, maybe metagenomics, you can learn a little bit more because you can get a bigger picture of like what other genes are present. And things like that, but you still may not be able to link it to any known organism that anybody's ever worked with in the laboratory. For chemicals, it's, you can do you can also do some non-targeted chemical analysis. It really depends on what technique you're using, but it sure is a lot easier to know what you're looking for. Also, yeah. It's definitely a possibility that a new thing can just get lost in all the noise of everything else that is just there.
So I'm also thinking about like sort of the synthesis and the optimization of, some of the methods that I've been hearing about today. Some of these for the first time again, so forgive my ignorance, but it sounds to me from what I've been hearing that, wastewater monitoring can at times be expensive because the hardware associated with it. It also sounds like there are opportunities for lower cost air monitoring sensors, but it seems like it's more difficult to detect viability through dust than it is through some of the other means that we talked about. So I'm wondering if there's like this global optimization, like, we monitor for this amount of time, but then if something pops up in the air, then we need to up the sampling rate for wastewater, or, like, how do these all play together to create systems for optimal detection?
I think that that's a really excellent question. And so I think that there might be different phases during times which of these are more important. So we probably have what I'll call sort of normal times. We're just chugging along, looking at something, and then maybe we see a trend we're concerned about. So maybe we ramp up a little bit. And I think it's going to be different in different areas as well. So it might be different on a college campus than in a prison or at a congregate care facility. I think that a lot of these cases, it'll be probably a lot of resource optimization about like what can we get away with in terms of this, and then what do we need to ramp up to if we start to see a concerning signal as well. And so I think the biggest question is, what is the question? In each case, what is the question that we really want to know at each time period? And then we have a lot of tools available to us, especially at this point in time, that we can actually target our analysis and our samples to that specific question.
Yeah, I definitely agree with Karen's point. I think the most important question is, what is the question you wanted to ask and you wanted to have the answer for, right? So like the surveillance can help you see the trend. And what I do is not really surveillance. I'm trying to prove that the aerosol plays a role in the transmission. So, viability becomes very important, not just detection as an example, right? And I use that tool to sample in a residential space versus the healthcare facilities. Again, that's not really to observe just the trend in each location, but in terms of risk at each very granular location, what is the risk among it? So depending on what you wanted to ask, you design the methodology accordingly to answer those questions. And each one has its own strengths and limitations.
Maybe in this field, do we have data set where we can go back, have a baseline, like wastewater surveillance before COVID? I mean, are there samples that have been collected that you can go back and have a baseline? I mean, is there such a thing?
So I would say for dust, we did actually go back and try to measure SARS-CoV-2 RNA and dust from pre-pandemic. Fortunately, we didn't detect any, right? Otherwise, would have been concerning. But I think I feel like for some of these things, we're almost at a new baseline normal now. For like it, almost the baseline data is probably more important now, prior to that whatever happened prior to the pandemic, as like, you know, regarding SARS-CoV-2 especially, of course, but even other things, a lot of things have changed. And I think that all could contribute to what that baseline looks like.
My answer would be, say, you got to have some samples you can reanalyze, right? Because SARS-CoV-2 was not known to us before COVID, and therefore definitely there was no analysis of SARS-CoV-2 before that. So if there are some samples that are preserved, then we may go back. So that's maybe another study we can look into. The challenge is, where do we have these samples, right?
I chime in here. I think there's interesting opportunities for like clinical samples of urine and feces or stool. I found one interesting paper on urine collected in Italy, and they were actually, this was archived for another purpose, for another disease. And they actually went back and looked for SARS-CoV-2 RNA in these urine samples, and they actually found, I think, 3 or 4 positive urine samples as early as like November of 19 in Italy. So it might not be wastewater per se, but you maybe could find archived samples of the things that you might expect to end up in wastewater.
Well, and one of the challenges we ran into is that the methodology, like if you just froze wastewater samples, then the viruses like were not recoverable. So wastewater alone doesn't work. You would have had to actually have to concentrate, extract, and then preserve the RNA, I think, to actually look back. But is the methodology that was used before the same as some of the optimized methods now, with some of the challenges in terms of being able to look back at these older samples?
Yeah, I was just going to add, and I think this feeds with what Aaron was talking about. When we look at built environment samples and dust, you're right? We don't have the databases, we don't have the libraries of the microbes, but when we look at gut microbiome, we do have the libraries. And so to your question about what is it that we should be screening, it may not be that the first place we go is where we don't know 99% of what is actually there, and going to more of the clinical samples or something that is much better characterized may actually be where we should be looking in the first step. So we really need to work on that intersection much more, which we don't usually do.
Any final thoughts? Other questions or comments? Fantastic, man. I want to thank both of you for your remarks and for engaging in this excellent dialogue as well. Thank you so much. Appreciate it.
Thank you.
And I'll take a quick second to transition up here to the front of the room, and then we'll dive into the next portion of our discussion. So we're going to jump into our facilitated discussion for top 3. And we're going to record the conversation because that's important to make sure that we get good documentation. Okay, so the questions for the facilitated discussion are going to be very similar to some of the questions that we asked earlier. Just our focus is going to be just a bit different, as this session is focused more specifically on what it is that we understand fundamentally about disease transmission in the indoor built environment. We've seen some good examples of different projects that have been able to shed light on some of those fundamentals. So we have a similar set of questions. So we're going to start again by just exploring what is it that we know regarding how disease transmission works in indoor environments. So, and it's okay if the takeaways, the conclusions seem to be pretty obvious based on the conversation that we just had. This is meant to help to be a summary, so it's okay. Starting off, what is it that we know that we know?
I'll lead off, Aaron, LSU. I think we know that this is a mechanistic process. And so there's from an emitter, the entry of some infectious agent into the built environment, it's transported through space and time and hits a receptor And we know that all this is mediated by physical processes. I think that's the best way I know to say it for now.
Appreciate it. Please.
I'd say generally we know that transmission in indoor spaces is much higher than outdoor spaces, as like one key factor to also focus on.
We also know, I think we know that from a standpoint of controlling things, it's best to block it at the source.
Yeah.
Mahinda from Clarkson. I think like the inhalation exposure is 1,000 times higher than the outdoor environment.
Sorry, do we know, I mean, doesn't that depend, that depends on a lot. I don't know who said that, but I'm just a little worried, like, that's not always the case. Is that just overall integrating all? I just don't want us to say something that's not.
Piece of information right now, but I do have in my presentation. I'll just look into it and then get back to you.
Okay, yeah, I guess as I say, I think it can be. It depends on the ventilation and the conditions. There's just a lot of factors. It could be, if the CO2 levels are the same as they are outdoors, it's going to be far less than that.
Yeah.
Okay, please.
Just a question. Are we focusing this conversation on respiratory risks, or are we thinking about others as well? And does it matter to people? Just inhalation hazards and respiratory pathogens, or are we also thinking about other sources of transmission in the indoor environment?
So I think, yeah,
Okay.
I would say all of the above.
Thanks.
Well, if I could add, so there is some disease spread in the indoor environment called Legionnaire's disease. So it's kind of tricky because it's not exactly waterborne, because waterborne people think of as being enteric, but it is waterborne in that it grows in the biofilms of pipes, and then it does cause a respiratory illness. It causes a very severe and
You're getting a little faint in the microphone.
Legionnaire's disease causes a deadly form of pneumonia. So in that sense, it's a respiratory illness, but it's not spread person to person. It's spread by the drinking water itself being contaminated with the bacteria, which are growing in the biofilms of the shower head. So I hope we can keep Legionnaire's disease as part of this discussion. Yeah.
Any other things that we know?
May I add to the first point? I think it's not just the physical processes. There are also chemical processes, and definitely, for the microbes, there are biological processes over there.
Yeah, and I'll just add to that the individual matters, but not everybody is going to be infected in the same way.
Okay, does that about sum it up? This is the known science. If you were teaching a course on the factual basis for our understanding of disease transmission in the indoor environment, are there other things that you would include in the curriculum?
I think this was maybe kind of said, but just making sure, remember that there's decay processes, right? So there's a source, there's decay, and then there's transmission routes, and also dilution. I mean, that's like the classic routes of transmission to think about.
I'm just going here, source, decay, transmission routes, and...
But like mixing and dilution also,
dilution, okay.
and thinking about concentrations that are received.
If I may, I think the human factors, the culture, social norms, all these also play into the role in the transmission.
Absolutely.
And we know some of them.
I think you're also missing infrastructure. So how the building is constructed, how the HVAC system is operated, what institutional cleaning processes are going on is also important.
Nicole Rocky, Duke University. I'll also add that behaviors are very important.
Awesome.
Just kind of a question. Do we want to include zoonotic or animals that might be in the built environment, or is that outside the scope of this?
It's a good question. I'm going to keep looking to NSF concerning scope.
Companion animals are a big deal.
Okay. Yeah.
Yeah, I remember in the COVID time, in the beginning of the COVID time, the cats were the first indicator who get sick compared to other types of animals.
So in that spirit, I think when we think about the foodborne pathogens may also transmit within the indoor environment. You think about your kitchen and cutting board, that could be a source of some of the pathogen transmission in the indoor environment as well.
I think maybe one thing that we know is that intervention has to be, multi-layered. Just because they're, building on the idea that there are many pathways in play, that it's, the idea that you're just going to do this one thing and that's going to solve the issue is, I think pretty well debunked.
Sounds quite thorough. All right, so then our next question is, what are the aspects of the fundamental science that we don't know? What is it that we still need, or where do we need to evolve our understanding? What are the big open questions that still remain?
I guess picking up on the conversation that we had earlier, there's a lot of unknowns, so we need to expand just our basic databases and understanding who's actually there and characterizing them.
I think the environmental factors influencing the transport and the viability of the microbes is very important, including in the air and also in water, probably also in soil or indoor environment in dust. So these environmental factors like temperature, humidity, all these things.
And I'll add to that, and understanding what structures or components of these microbes cause them to be more or less viable in the environment.
Please.
I'd like to add a non-microbiology component to this discussion. We know that, for instance, if you're exposed to outdoor dust, you might have an increased susceptibility to infection because the dust gets in your throat, causes some irritation there, which allows an increased likelihood of infection. So abiotic factors might influence the susceptibility of individuals to these pathogens. So even though we're considering only the indoor environment, susceptibility of the people in that environment because of other things that they encounter, whether that's organics, VOCs from the carpet causing increased susceptibility, or they came in from outdoors and outdoors has higher dust issues or other air pollutants that might cause an increased likelihood of infection.
Gotcha.
Building infrastructure that can act as a source or reservoir for pathogens. It's probably more with the bacterial pathogens than the viral, and how to manage that infrastructure to help mitigate or reduce the risk. of those organisms? Thinking about Legionella and others like it.
Aside from abiotic factors, I think the biological factors, let's, for example, when you have two viruses at the same time, who contributed to what, how much is important. And then similarly, if you have infections by bacteria, and we also have the SARS-CoV-2 or flu virus, then it's not 1 + 1, it's maybe 1 + 1 = 10.
Yeah, so to add on to that, the comorbidity or co-infection is incredibly frequently observed. They often, like when they have gastroenteritis, they say, oh, it's because of X organism, but also it's this bacteria, right? But they often also have this, not just a C. diff infection, but like some other viral infection is going on as well. Yeah. So co-infections are interesting.
I think like relative contribution of various pathways. So we know there's a lot of pathways in place, but when you're designing, remediation and intervention strategies, which pathways do you target, which is going to change by, the relevant pathogen. I think of like norovirus on cruise ships, right? Where do you do something and how and why? And then can you, generalize that to other settings is something I think of.
I think that there are things we know about ventilation and things that we still don't. So I think that there's a lot that we could still learn about. There's actually not that much data in the literature directly linking different ventilation strategies to actual case numbers. And I think that I know that that's a health outcome, but that would definitely be helpful. And just as I said, there's some things we know about ventilation and some things that we still don't.
I think another thing I would add here is, how humans interact with their built environment. How does the design influence that interaction? Are there designs that, nudge or produce behaviors that might be better in terms of preventing infectious disease? So how can, like, instead of being so dependent on individualization for like, hey, everyone wear your mask, is there a way that our built environment just passively adapts for us based on its design?
Yeah, that's a really good. I often say when it comes to the sustainability of most things, in the end, it may come down to architecture. Right? Like, can we make this a common aspect of how people just interact with the built environment? So that resonates. I appreciate
it. Something that came up in the panel or the session before was the question of potential negative side effects of disinfection. This sort of combines in microbiomes too. And so there's good bacteria and good microorganisms and bad microorganisms. And if you're disinfecting things and surfaces, is there a chance that you're creating risks because you don't have maybe protective effects of the rest of the community?
If I can, go ahead. Just to jump in and you can add more, I'm sure. So there is concern that the disinfectants could select for antibiotic resistant bacteria, for example. And actually this whole discussion of whether it's airborne versus surface, it's very important because, these are chemicals and why overuse them if they're not doing anything for us. So it's very important to determine the benefits of disinfectants and optimize their use. They are very important in the right context. And then related to that, I think Karen D highlighted, there are a lot of microbes that are just there, right? But what are they doing there? And are there beneficial ones we don't know about? So I think getting a better handle on what are some microbial indicators of a healthy environment. Yeah, just to chime in on the, yeah, I think potential side effects is, there's a paper, actually, a review called Unintended Consequences of Cleaning Your Air. And when people spray chemicals in the air, they often make the air worse, not better. And so, yeah, that I'm on it. We have there will be a report coming out from National Academy of Sciences and Engineering that I helped write over the last 3 years. And it's all about, what's the best thing to do, because even like one of the real downsides of getting to the sort of cleaning the air in a built environment is people will spray chemicals, they say, which make things worse. They can create an heavily oxidizing environment. The other thing they're doing, and I don't know where this would fit, and this is has been a real problem, is, the best thing to do is just to filter it. It's like, that's what we do with our water, just filter it, get it out if you can. But nobody knows what HEPA filters, nobody knows what filtration to use. And so there's been all these snake oil people that have sold ionizers and fancy gadgets. They have all these super Hector HEPA, all these fancy names, which just means the price goes up accordingly to schools and stuff. Their guidance has been horrible. And so people are always thinking by paying more, you're doing more. But in fact, in these cases, there's very little known about. It's the simple, almost in this case, the simpler, the better.
So I guess feeding off of that and going back to the databases, I mean, we need better functional microbial ecological models to actually be able to answer those types of questions.
I wanted to jump off of that as well as what Amy said in terms of just also an understanding of those beneficial microbes. I think that there's been less work in that space, and we've started to see indications that some of them are beneficial. And so just better understanding who those are, what they're doing, why they're beneficial, and how we can keep them there doing what they need to be doing while we take care of the ones we need to get rid of.
So I'd say building off of that question, I believe our next question is, yes. Please.
Let's say wastewater. Can you go from a sequence? You guys have done some nice you go from the sequence for giving structure and you know how.
Yeah, so the question, so just to make sure I got the question right, so you're saying, can we go from a sequence to like an aerosol structure, how it's
Exactly. For the sequence, I mean, that's the biology. What's creating
right, and what the payload is. Yeah, so from the sequence, we do have usually fairly limited data, and it depends on what the sequence is. So a lot of times it is just one amplicon that we can kind of read like a barcode a little bit to say, hey, it looks like this particular species was present, and here's where it was. If you do more in-depth things like metagenomic sequencing, or you can do even metatranscriptomic sequencing to look at gene expression, you can start to get an idea of something like function. But a lot of times, like I was saying before, we don't know who all the sequences belong to. Like a lot of them we can identify, some of them we can't. And so it's difficult, especially if you're working with just amplicons, to get a lot more data out of that beyond who's there. I don't know if did that answer your question?
Yeah, I mean,
yeah, no, so that's the work that I think Romy Amaro has been doing some of that. But if you can figure out what it is, like a family, like actinobacteria or some type, one of the things we did, we were able to say, okay, this type comes out of the ocean. We look back what it was, figured out that the cell walls have these mycolic acids, which are waxy substances. So it made sense, right? So you can kind of do that. And then Romy's models, she's doing these billion atom simulations, but that's that's not just the virus, or you'd have to know which virus you had. And then she's mixing that with like mucins and other respiratory fluid, and then trying to see, what happens as these things evaporate, and sort of looking at sort of predictions of how stable they are inside of the droplets, because that's the hard part, is they're not just little naked things floating around, right? You have to kind of take into account all of the interactions. And that's the part that there needs to be a lot of work done on, I think, still.
I'm curious, how computationally intense are those investigations?
It's intense. It's intense. Luckily, NSF, we get lots of time, but she's using computers everywhere for her stuff. And, we're looking when we do when she does an aerosol particle and how fast things redistribute chemically, it's like she can do like what happens in the first 400 nanoseconds. That's it. Like that uses up all your time. So it's pretty crazy.
And I think it can really depend what you're doing. Like if you're looking at amplicons, you could run that on a normal computer. But if you're going to do like metatranscriptomics, like we often use the Ohio supercomputer on their huge memory node. And, we work with them fairly closely to make sure we can, do what we need to do. So it really just depends.
I think this raises an interesting point around exposure science, right?
Yeah.
How do we conceptualize it? I mean, this little virus and you breathe it in, and what's the chance you get infected when actually these things are moving through the environment and not as discrete individuals, but as, potentially multiple particles at once. And there are different aspects of that enhance their persistence probably. And so I think there's been some, maybe a paper, I guess it's been a few years now, on norovirus and how it's like actually sometimes excreted as these little clumps of viruses instead of just individuals, and that affects how persistent it is.
Saying that the progress is like 30 times.
So that makes me curious about one of the earlier questions that was asked about the ability to extend some of these research methods to other areas of the world. And I'm wondering if there are implications for whether or not we're still connected to Zoom first, but also implications just for, I guess, an environmental justice perspective. How is it that we make these research methods accessible? Right? If we're globally impacted by disease, how are we globally benefiting or distributing the benefits of research? Maybe not a question we'll answer now, but Okay, so our next question here is about any aspects of research that have been sort of undervalued or ignored in the past. I was just thinking of one of the prior comments about beneficial microbes, for instance, I think was one of the ones that had just come up. So I'm wondering if there are other areas of potential research that haven't been as well explored in the past.
I will say we talk a lot about like influenza virus, SARS-CoV-2. There are a lot of other viruses out there that aren't as well studied that I think would be important to look at, and bacteria and other species.
So this is going to be a common theme from people who are in the last review session here. We also don't know when a patient goes to the hospital and is in a clinical setting, what is causing that respiratory disease. It is not often that the doctors are going to call for a panel to figure out it's caused by the latest influenza or whatever. So, and even to the point of we don't always know which ICD codes to pull associated with that disease that we're interested in. So a better collaboration with the clinical or medical side and better records and access to those records would also be helpful.
Absolutely. Thank you. That's a good point. I think it's some of those limitations on data collection, right? Where infinite data would be useful to the research community, right? But it's always a balance.
Sorry, we know that this community has an increased number of respiratory infections. They're checking the hospital, they're seeking medical care, but we don't know what's actually causing that disease. And therefore we can't go back into the built environment and say, oh yes, it's definitely coming from this source, right?
Right. Yeah.
There's many unknowns on what is making people ill in the community.
So what does a potential solution pathway look like in that context? Is it just enhanced communication and data sharing between the research community and the clinical community? Is that part of it, or does it go beyond that? And an open question, by the way.
The hospital is not going to pay for the test because it's expensive and it's not necessarily needed to provide clinical care to that patient.
Yes.
So they're going to treat the symptoms, not necessarily the particular disease causing organism in a respiratory case, right? Unless they maybe think it's antibiotic resistant or something, right? Now, what we could do is, as we develop more rapid sensors that are useful in the built environment to test for these organisms, maybe those can be modified to work in a clinical setting to make them faster. And then, but that's going to require like FDA approval or whoever the regulatory body is for that particular test. It doesn't mean that we shouldn't work on better, faster sensors and methods to detect these organisms, but we need to recognize that maybe we can't get funding because we can't say, oh yes, there's a huge outbreak of this particular pathogen every year, because we don't know. It's the bottom part of the iceberg that's under the water.
Yeah.
We know what's happening, but we don't know what's causing it, for instance.
Makes sense.
Sounds like a good place for some wastewater surveillance.
And sequencing and proving that it's viable.
Yeah, the same challenge. I remember when we were talking about fecal shedding, when we were talking about SARS-CoV-2 at the beginning, and like not knowing shedding rates and who was shedding and if they were. So I think a similar thing, we're taking a respiratory sample, but also fecal samples and figuring out, what's going on with patients clinically.
Interesting. Okay, so it sounds like we might have some alternative pathways to getting to that information without coercing the hospital into doing things that they may not want to do. That sounds promising. Gotcha. Okay.
I think one thing that's striking about the built environment to me is that we've mostly designed it for our own comfort. And now we're kind of recognizing that we've maximized for our comfort, and maybe that's also maximizing for disease transmission within these spaces. And so then there's also like energy considerations, right? In terms of air conditioning. So that's probably something that we're going to have to think a bit more about trade-offs.
Can you expand on that just a bit? Just the sort of the energy considerations for air conditioning. You're saying just like enhancing filtration could have.
Well, I think, we're learning more about humidity levels and temperature levels that enhance persistence. And so there might be opportunity. I guess what I'm thinking is like, are there situations where we have to accept maybe being not quite in our optimal comfort zone to decrease some risk, or we have to find energy efficient ways of making the space safe. Those are my kind of just initial thinking.
So, just quickly, so basically, I think this relates to what you're saying, but the tightness of the building. Right, so, and especially if you contrast Europe versus the US, then the US, we tend to have tight buildings where we very carefully dial in the temperature and the humidity that makes us all comfortable, but pathogens also like the conditions where humans are comfortable. And then there's not as much circulation with outside, whereas in Europe, you can go to a hotel in Europe and you can actually open the window. What a concept. Like, a lot of the buildings, it's required that they design that you can open the window and get that direct ventilation. But that's terrible for energy conservation.
It's
But
funny to,
that's actually going to change under future climate scenarios, right? We're seeing many different communities where they actually need to implement air conditioning to prevent, heat-related stress or heat deaths, right? So you might then predict that there could be more respiratory diseases surviving in those communities, causing an increased number of outbreaks.
would you mind repeating that? I just, the microphone, I didn't click.
Yeah, I'm sorry. So basically I'm saying climate change is going to make places hotter. They're going to need to use more air conditioning to prevent heat stress, heat-related deaths. That may increase the number of outbreaks of these kinds of respiratory diseases if you follow the assumption that the conditions that we make indoors for air conditioning causes a long-term survival and or transmission of those pathogens.
Gotcha.
I think, are you saying like for the AC, you'll just be recirculating your air, right? Because it's not energy efficient to bring in fresh air, right? Right. Right. Yeah. Yeah, I mean,
I'd say, okay. I was going to recap. So I think part of the conversation was just the recirculation of air and the ability to create more persistence. So we're getting more situations where we're not bringing in as much fresh air,
right.
And that trend is globalizing because of climate change and the need for more HVAC infrastructure.
There's a battle between, just cooling things off, but then there's an energy efficiency. The easiest thing to do is to bring in fresh air. That's the easiest thing, but you can't do that anymore, because it is to say you're bringing in either hot air or wildfire laden air. You're bringing in more polluted air. So now you're having to recirculate the air, and then that leads to more disease, I think.
So Emmanuel, actually, I
Yeah.
Yeah, the wildfires have just made it harder. We had an issue time where we were getting so much wildfire smoke that we were closing up our houses and setting off our smoke alarms in our houses. There was just so much smoke coming in. And so now you're sealing the house, you're reducing ventilation, and more people. So we had to choose COVID or wildfire. Like, you're sort of being forced to choose.
Kind of crazy too, being on the energy side of things. It's my background. Looking at things like passive house design, where it's like, let's make the envelope as tight as possible, double all of the insulation, and control every bit of air that's exchanged from the external environment. Like, wow, yeah, I think we need more interdisciplinary work when it comes to designing for energy efficiency. That's pretty bad.
And there was a comment in the chat about fungi and, thinking about increased flooding and all of the risks associated with that. So in that case, right, opening your windows isn't going to help. That's where you want air conditioning.
Gotcha.
Be able to lower your humidity.
It's also where you absolutely have to have filtration because, sometimes people really fixate on ventilation and that's not going to help a lot of times in these cases. You need filtration too.
Gotcha. All right, so I'd say as somebody with a very little background before coming into this conversation, but also like somebody who has a PhD and is also had a research career, I would venture to say that the average citizen is not very prepared to understand the dynamics of climate change impacts and how to change or be responsive in their behavior in the way that they're managing a residential environment in order to optimize their health and safety. And I'd extend that to say, as somebody who's also lived mostly in urban environments and rental units for most of my life, that a lot of people don't have the agency to be responsive either. So, this definitely sounds like a very interesting public health challenge.
I'd like to throw out another potential undervalued thing, and that's that we're not rational.
Yeah.
And it seems like in that case, like this idea that we give people more information and they make better decisions just doesn't work.
Sure. Yeah.
And so we have to account for that in our engineering and interventions.
Going off on the climate change impacts, I'm happy that mold and flooding was brought up. I think we're going to see more and more and more and more of that, unfortunately. I wanted to add also water stress leading to probably more need to store water, and also having water saving fixtures. And so those can both result in opportunistic pathogens having an opportunity to come into the home.
Can you clarify the connection there between, say, water saving fixtures and?
Yeah, so this is something that probably Amy can better talk about, but like Legionella, like a big hypothesis and people have shown that was like water saving fixtures, you have more stagnation, so you have more opportunity for Legionella to either grow in biofilms or be able to partition out of biofilms into water.
Okay.
And more reservoirs. So there's definitely a trade-off between potentially water saving features and also set points on how we're the temperatures at which we're storing water and have water circulating through our systems, which is an energy related question. So those things are important. And then from water storage tanks have opportunities. And I know that there's, certain municipalities that because of water stress, they've like cut the municipalities off from the water network systems. They're tanking water into their homes and storing. And this is something that happens around the world. And if we want to talk about environmental justice or other locations, stored water is most people around the world are storing water, but we're going to see more of it in the United States in the years to come.
Gotcha. Interesting. Okay, so to be clear, because again, just hearing this for the first time, so like those reduced, shower heads that reduce flow in order to save water. So you're saying that by lessening the rate of flow of the water, there's a potential for more buildup within the pipes, and that can harbor. That's terrible.
Happy to elaborate. No, I'm so glad you brought this up. So there's a combination. So with less water use, then there's more stagnation.
Yeah.
That's one. So that means the water is sitting longer, and just like anything that sits longer, there's more opportunity for microbial growth. Like think of your milk in the refrigerator, right?
Sure. Yeah.
Then there's the issue of the velocity of flow.
Yeah.
Right. And the shower head design influences that in ways that are barely studied to my knowledge. But, and the hypothesis can go either way. Like maybe high flow is good because the velocity can shear biofilms and kind of clean up the pipe, sweep out sediments and things where microbes can be hiding. But it's also bad for the same reasons because it's sweeping out microbes into the aerosols that you're breathing in the shower. And so I think there's a lot of fine tuning to be done on, what is the ideal water age, stagnation flow? And yes, there's push to reduce water use in green buildings. I think this building we're in right now is a green building. I noticed when we entered, which is great, but not all of these things are really thought through and optimized.
Thank you. It's crazy, just like, things I've never thought about, but then as soon as we talk about them, it becomes immediately obvious. And it's like, oh, why haven't I thought about this?
It's our job to keep you up at night.
Thank you. Thank you. I'm so glad that I was invited here today.
I think another point too is the temperature of water. So I live in Baton Rouge, Louisiana, and in the summer, I mean, the water's coming out of our tap at 90 Fahrenheit from the municipal supply. So there's issues around residual chlorine and these increased temperatures and, what that's selecting for in our drinking water supply. So, and that's going to become more and more of an issue as climate change progresses.
I used to live in the Middle East, and the hot and cold water would switch at different times of the year. So, crazy.
Well, if we're going down this route, we should also talk about water reuse. And if your water is toilet flushing water is like gray water versus drinking water, and the potential exposure routes of water that is not treated to potable quality, but is still within the home environment.
I gotta hang out with y'all more. I got a lot to learn. I'm a big gray water and reuse fan, and I have like off-grid property and stuff. And now I need to know if I'm doing something wrong. We have to talk more. Okay.
I think another issue in terms of climate change is a lot of rural communities, right? You have decentralized wastewater infrastructure paired with private wells. And so, you have flood events and potential, hydraulic connection between this wastewater tank that's in your backyard and your drinking water supply. And I know that's not necessarily indoor built environment, but these are built artifacts. So especially in rural communities, subject to these flood events, that's going to become an issue. And we're increasingly skeptical of groundwater, right, in terms of microbial safety.
Yeah.
Sorry, can you use the mic?
Okay. So human behavior, part of the workshop was human aspect. So can you guys comment more on the human side of things?
I think so I've done a bit of work on household water treatment devices. And we know that, when you lose 0.1% of adherence in terms of using that device consistently and correctly, you forfeit a large amount of the health gains associated with it. And so we know that human behavior modulates how effective our interventions are. And we know that we can design things to be either more or less user-friendly. Now, how well that translates to something like indoor air quality and masking and things like that, I think is an open question. But human behavior is like a critical component of the engineered system as the end user. And we know that basically we can subvert all of our best intentions that are designed in with irrational behavior, let's call it.
Yeah, so the Brita water filter that my aunt hasn't changed in 3 years, for instance, is likely doing more harm than good at this point.
Related to the human behavior, if you look at the indoor microbiome in general, and I'm not talking necessarily specifically about pathogens, but just the microbes in indoor spaces, each indoor space is so unique. If you look at all the different aspects that might influence it and the variability explained by any of these factors, they're all very, very small, because each indoor space is influenced by so many decisions. Do you take your shoes off before you enter the living space? What is the ventilation like? How many people live there? Do you have pets? What types? And the list just goes on. And so I think each individual space is actually so unique in terms of just the general background of microbes that are present, largely due to both building design choices as well as individual choices.
I think we need to have a community engaged research in order really really to make the outcome better. Because right now, a lot of time we dump the data to the community, but they do not necessarily want to accept, or even though we have good intentions. And at the same time, we are also missing the opportunity to incorporate their inputs to design better things that can be accepted by the larger community. So I think that's something you can say ignored or undervalued in terms of community engagement research.
Okay, so I'd like to move to our next question, and this is just around short-term opportunities. So we've talked a lot about, excuse me, some of the aspects of the science that are known and unknown. We've brought to light, for the first time for myself, and not for the first time for many of you, many challenges that do exist. And so I'm curious about how do we move forward, and what are the actions that you all believe make the most sense in the short term? So what should we do next?
I would say we should walk out of our environmental engineering community here and go to reach other communities to identify the skills that we need in order to solve the problem together.
Absolutely. Thank you.
So if I frame telling us to do research, is that what you're telling us?
I'm talking about, say, because NSF will fund scientific research, but not really medical. But for this case, we need to have medical collaborators to solve the problem. So that may be, say, NSF and NIH together as the example. Or say, if you wanted to work with, say, agriculture experts, then that may be USDA, that kind of collaborations, but not just funding agencies, really the scientific research community needed to come together. And very often it's hard to establish that, at least in the beginning, if you don't know the people.
I guess that would be part of my question, or part of a follow-up here is, who is missing from this discussion? Who are those collaborators that aren't present in conversations like this? So you mentioned the agricultural space, medical doctors. Are there others that, is it infrastructure operators or utilities or
Architects,
designers?
architects, behavioral scientists.
Also, those setting building regulations, like building code regs, really important.
I think Claudia in the morning actually has a good list of people we should have. Microbiologists, virologists, doctors. She can probably say better than I can.
Sure, I'd be happy to step in. Yeah, I mean, we need all the people that are building the models for the medical field, but applied to environmental. So yeah, the microbiologists, the engineers, the virologists, the biostatisticians, the bioinformaticians, but then also what was mentioned with the people that are actually architecting and operating the building, all the way to the policymakers that will help us derive it. So I've got a slide that's got all of them listed from this morning.
And epidemiologists.
Social workers.
So Emmanuel, I'm going to jump in here. Say, when I look at these, I mean, some of these are long term, right? I mean, what short term, meaning like maybe you can submit a proposal to NSF in 3 years, 5 years, you can make some progress. I mean, which exactly among these you could think of a 3 to 5 year type of project?
It's a great question.
I guess I'll keep going back to the databases. We just need to build our actual infrastructure for organisms so that we can start getting at those questions, not only just from the amplicon data, but so we can look at metagenomics and metabolomics so that we can actually develop the sensors and get a better idea of who is there so we can advance some of these questions.
Mama, do I want to go back to your idea? Maybe we should go back to past samples to do more analysis of these viruses which were not analyzed before.
There's been a lot of discussion of indicator organisms and model organisms and surrogates. And I think it's important to remember that, one surrogate will not necessarily meet all of our needs for a specific organism, and a lot of it's process-based. So, if you're interested in SARS-CoV-2, for example, one surrogate may help you understand when it's exposed to this thing, but a different surrogate may help you understand when it's exposed to that thing. And I think that there's some really interesting work that's being done trying to better understand mechanisms of inactivation and removal and transport, and how, like, molecular mechanisms, which is where some of these partnerships with microbiologists may help. Because if we can better understand sort of those molecular mechanisms, we may be able to better find the best surrogate for that process, right? Something that, another organism that shares a similar characteristic. So I think that some people are doing that kind of work, and I think more of it is definitely beneficial.
And I'll just add one word of caution for going back to past samples. A lot of the environments we work in are low biomass. And so that means there's increased risk of contamination. And so just for our community to learn from the fetal microbiome community that ran into issues related to low biomass samples. And so I think we need to think carefully about how those samples were collected to make sure that when we are analyzing them, we are not just getting the contaminants.
The final question that I'll ask here is, are there any challenges that we anticipate, like the one that you just brought up?
I think going back one point, maybe an opportunity near term is like some kind of low fidelity modeling exercises to kind of, I mean, we have a feel for our mechanical systems, our plumbing systems, our fomites, things like that. So there's probably room for some just like model-based hypothesis testing in terms of, you know, where can we intervene maybe? And then in terms of challenges, right, there's just the data to populate those models are extremely sparse. So dealing with the uncertainty in building those models and using them to kind of maybe derive some more fundamental opportunities for investigation.
Thank you.
Someone said this earlier, but I think it belongs here, which is that every built environment is different. And there's probably many, many factors that contribute. And so in order to tease apart all those factors, it either needs to be very controlled or have a lot of data, or both.
We talked about some of the facilities and resources, equipment, computing, things like that that are needed for some of the research. Are there any other sort of resource needs? I know we've already mentioned funding, right? So we already we know about funding, but any other resources or services? Are there particular user facilities or capabilities that need to be more broadly accessible?
Well, something we talked about in the morning session was if facilities that have BSL-3 or BSL-4 capabilities were available to people for experiments. Yeah, just offering the opportunity for actual pathogen work to be done.
And I'll just throw out a plug for our test bed through our engineering research center. So we do have access to a test home on the Duke campus, which we're currently renovating, but thinking about it similar to the NIST test bed, where we can test more chemical and physical types of exposure, but thinking about it from a biological perspective. So just common test beds that we can work on to help us benefit from our data.
Awesome. Awesome.
Also, someone has brought up databases a bunch of times. I think a challenge and something that we need is a highly curated, accurate database. And I know NCBI is doing a lot of work on this, but also the databases are contaminated and misannotated, and that affects all of our research.
So Emmanuel, to just jump in here, to challenge sometime, can you comment on infrastructure and pretty much like infrastructure? For example, NSF had what we call mid-scale infrastructure. So do you think this community needs a mid-scale infrastructure when I hear BSL-3, BSL-4 type?
I mean, it seems like, mid-scale is, remind me, it's 4 to 50 million?
Yeah.
Yeah.
Mid-scale can go anywhere from 5 to 20 million type. Less than 100 million. So because NSL have these large facilities that are above 100 million. So in that range, yes.
I just wanted to add that I know that would have definitely helped us when we were looking to study if, the viability on dust. At the time, we couldn't get into the BSL-3 facilities. And so I think there are a lot of questions that could be answered only in those facilities. And they've largely been more reserved for the medical community, which,
make sense, but I think that this community could absolutely benefit from the ability to do those experiments.
I think as well, I mean, building off Claudia's point, these test beds, right? I mean, those are very unique opportunities. And so, one in North Carolina, I mean, based on what we know, right? I mean, you would expect, like, depending upon where that test bed is located and the climate there, that's going to affect what you might observe. And so those types of controlled environments, so to speak, I would think would be really useful for understanding some of these fundamental questions.
Yeah, it would be cool if there were like apartments purchased in places around the US that have been like in old buildings, like buildings that have been used for 20, 30, 40 years, right? Like those are more representative than anything we can do in the lab, or if you buy a new house or whatever. It'd be super cool to have the opportunity to do that type of sort of infrastructure work in a building that's been used in different places.
So one of the things that we discussed was even intersection with FEMA and houses that, have been condemned. So thinking about a way to be able, with locations where we don't want people to live, this could be opportunities for our community. Yeah. Goes back to what you were talking about, somebody's dirt is somebody else's treasure, or whatever you said.
Then I want to say thank you to everybody for the discussion. And for leaning on your experience to help to inform this important conversation. So we're going to take a break.